physicists, there's venues for biologists, but there's only a few cases in which really the community come together. And you'll see that today with my talk, we'll shift a little bit from the facts of biology to the sort of the theories of physics. So we shift from who and what to how. And of course, something we gain, something we lose in terms of accuracy. And so Ed has the date is actually wrong, sorry. Ed has asked me to give an introductory talk, so I'm gonna give my best to give a little bit of a more pedagogical talk. So the title is Genomic Energy Landscapes. And why is the physics community very interested in this? The physics and biophysics community, but mostly why the theoretical physicists are interested in this? Well, because it's an extremely interesting system. Typically, we see DNA. Of course, you know all these things. I just want to give my perspective in this. So DNA is seen as a storage device. And I call it the partial dogma of molecular biology. And we know this is partial because we know there's epigenetics, so there really is a feedback. It's not one way down from DNA to proteins. But you know, what you know, but what the community is starting to learn now, is that there's more than that. Because DNA is not just a storage device, but it's also a machine that processes the information on that is stored on this long tape, which is DNA. So DNA, you know, the structure of DNA in the nucleus participate in this information processing of inputs from outside the cells, which can get through the cytoplasm and get processed by proteins, and then it gets to the nucleus, and then in the nucleus, a combination of DNA and proteins keep on processing this information. So it's a machine. And at least in my view, it's a machine. And in that, it's very, very interesting from the point of view of a theoretical physicist. And as a machine, we've been knowing in molecular biology that machines, their structure and their functions are related. And this is not, again, this is not novelty. We knew that the structure of DNA along the cell cycle, for example, it's very much related to what it needs to do so that the mitotic chromosomes are sort of optimized to allow the separation of the genetic information. So we know that the structure changes along cell cycle. We know that that's related to its function. We're also starting to see that the structure changes along differentiation. It's another function, another added layer of complication, and it's related to the structure. Of course, that's not just structure, there's many factors, but structure is one of them. And even more recently, we're starting to see that the structure changes along evolution, and that's another clock, another time scale. So we have 3 different time scales. We have time scale of the cell cycle, we have time scale of cell differentiation, and we have a time scale of the evolution. 3 time scales, changes in structure that are related to function and to other things. And of course, you know, I don't need to introduce high C. This is classic slides. You probably have seen it already 10 times during this meeting. So high C really revolutionized the field because it gave the theoretical, it gave a possibility for the dry lab to really do things with a data-rich data set accessible through public databases. And of course, high C measures the frequency of ligations between 2 loci I and J. So if we want to use IC, first thing we need to understand what that means. And at least from a point, I'm talking about from a point of view of a physicist, which is interested in statistical mechanics. So we need to do sort of like we need to introduce a mathematical formulation of what that means. And that goes first understanding that you can do ideally you can have one chromosome in one conformation and do one experiment. And in that case, the result of your ligation assay would be binary. It would be like either two loci ligates or they don't. But then what happens when you do multiple experiments on the same conformation, or you do one experiment on many different conformations? Now, these two things are related to, from a theorem, is like, it's called the ergodic hypothesis, but it's sort of like pretty sound hypothesis. So this situation, which one chromosome, one conformation, 100 experiments, is the same of one chromosome, 100 conformation, one experiments, right? That's an assumption, which typically works. Now, in that case, the result of your assay is not yes or no, but it gives you a probability. Given one conformation, you have a certain Cartesian distance between the two loci. Given that Cartesian distance for the two loci, you have a certain efficiency of that ligation. So it's going to ligate, let's say, 0.3 times. 0.3, 30% of the time that ligation is going to happen. So this function f maps Cartesian distance to frequencies. That's essentially frequency ligation as a function of a Cartesian distance. Now, how does that look? I mean, clearly, if you're very far, you're not going to ligate. And if you're very close, you have high probability of ligation. And in the middle, we don't quite know very well. So what is the exact shape of this function? What is the distance at which it goes down? Is still sort of an open question in the field. My lab is working on that. I'm not going to talk about that. But it is an important question. But all in all, the two extremes are clear, and there's going to be some transition in the middle. Now that it's when you do 100 experiments on one conformation, because one conformation is going to give you one Cartesian distance for the two loci. But then now we know, at that time we postulated that there are many different conformations in that ensemble. So now what we need to do, we need to take this and average it over the distribution of the conformations. Many different conformations, each conformation comes with its own probability, and that average is what we see in the Ising map. Now, people who know a little bit of StatMech in the room, they know that typically that probability distribution, we write it as the exponential of the energy. This is the canonical ensemble. It's a pretty standard approach. So essentially here, if we want to learn something from this experiment, that's meaning that we are observing these probabilities, and we have postulated a certain shape for this function, and now we want to know what are the structures that are in that ensemble. We want to know what is the probability associated with each one of these structures, which is this probability distribution pi, which in turn is this exponential of the free energy U. So this was our approach. So it was a rather ambitious kind of task to pick up. We wanted to create a physical theory for chromosome folding. And a physical theory is constrained by what we know about physics. It needs to satisfy the law of thermodynamics. It needs to avoid spooky action at a distance. So there's a certain number of constraints that come in straight from just basic physics. And then there's other constraints that comes out from experimental observation. So through the years, this theory developed as a theory in two steps. The first step is done with some machine learning methods, which we call megabase. And the second step is done with physical simulation with an energy function, which I will introduce now, which is called the minimal chromatin model. The theory takes as an input epigenetic information, which you can get from, say, the ENCODE database, and gives us an output 3D structures. And then you're welcome to do whatever you want with those 3D structures. So it's better to start from the physical simulations to start to understand this pipeline. So this is what we're going to do in the next couple of minutes. So at that time, in 2015, while I was working on this project, the ansatz was the following. I come from, protein field, molecules. In proteins you have amino acids, and the amino acids, they encode the fold. And then you have some strong contacts that they also collaborate with keeping the fold stable. So the ansatz there, the hypothesis was that the structure of the chromosome is encoded by some sequence, which at that time I had called chromatin structural types, and the CTCF was something that was also started to be known. That could create some stronger loops. So that was the ansatz. How do you take these ansatz and put it into formulas, and then the formulas into code, and then the code into results? So this first ansatz is that there's phase separation of chromatin structural types. If and when two loci, they meet, when they meet, they carry certain energy that depends on the chromatin structural types. This is well known in physics. It's been called block copolymer, it's phase separation. In this case, it was quite clear that that sequence was not the DNA sequence, but it was the epigenetic sequence, because in the 2014 paper from Aiden Lab, you could see, for example, this nice alignment of the compartment patterns with epigenetic markers. And that sort of like also accommodate the fact that you can have different structures in different cell lines due to changes in the marking patterns. For example, here, these are two human cell lines, IMR9G, M1278, and you can see that there's a change in epigenetic markings, and you can see that there's an associated change in the patterns, which you have dark and then light. Here you have light, dark, light, dark. So it was quite clear at this point. This was already also in the air. I mean, there was some work from Cavalli's lab, and some work from with Daniel Jost, and some work from Mariano Barberi, also going this direction. So that's where the idea came to me. So phase separation. And the second term, well, the second term is a little bit more controversial. At that time, my working assumption in my mind when I was doing it is that the second term was defined as the rest. So there's some stuff that is phase separation, and there's some stuff that I'm going to call the rest. And how does the rest look like? The rest look like as every time two loci they come into contact, they carry an energy that depends on the genomic distance between the two contacts. Now this is rather, in a sense, it's rather unusual, and it found a tremendous amount of pushback from the polymer physicists, because they were saying, with some point, I mean, they had a point. They were saying, how does a loci knows how long is the loop formed by that contact? Because a loci is only, a few nanometers across, and the loop can be centimeters. And how does that know? Where's the memory? They had a point. It's rather unusual in polymer physics as a term. On the other hand, as we've seen, there's a very specific slope in the contact probability that you might want to reproduce. And since this depends on genomic distance, the ideal thing is to just throw in the term that depends on genomic distance. So that's one way you can see it. Another way you can see it is that if you come from the molecular world, there's a long history of energy terms that depends on the distance. For example, this is Linus Pauling illustrating the alpha helix. The alpha helix is formed by a preferential energy in contacts between I and I+4. It's a translational invariant energy term that depends on the polymer distance. In this case, it's not genomic distance, it's the amino acid distance. So I+4 give you a helix. So there's a long history of these energy terms in the field of molecules, but they're rather unusual in the field of polymers. Then it became clear, because at about the same time, folks were working on these extrusion models. So this is actually what is being done before. But then about the same time we were publishing our paper, they were publishing their papers on extrusion, and then it all became clear. The memory of this distance, this length of the loop, is coming from the proteins. So the products have been extruding, and they know how long they've been extruding. So there's a specific profile of loops that are formed by molecules, and that profile depends on which are the extruders, how much depth are there. You learned a lot of this yesterday. So it all became kind of a lot more clear, and things have been discovered. And now this is sort of like a lot less controversial. But the interesting thing is that this was discovered from the data. And extrusion has been discovered in the meanwhile, but the shape of this energy function, which we call the ideal chromosome, it hasn't changed. The shape of the ideal chromosome hasn't changed since 2016, because it's in the data. It is what it is. So these two answers, they give you an energy function, this Hamiltonian, and this Hamiltonian is the Hamiltonian of the minimal chromatin model. And then when you go rationalize, this energy function really has two terms. It has phase separation, and phase separation only acts across space in Cartesian distances. And it has the ideal chromosome, which is due to the mother activity, and mothers acts along the polymer. So we have across space and along the polymer. No spooky action at a distance, so the energies, they only exist if the content is actually formed. So this is the energy function, and that's the theory essentially. And then that's the things you need to do, which is finding the parameters. Finding the parameters, it's a little bit tedious, but you can do it in 100 different ways, and they should give you the same results. And if your results depends on the way you do it, that's not good. So in our case, we did it. I mean, we did it with some Newton's iteration. Now they're doing it with this fancy algorithm of machine learning like Adam. But it doesn't matter. It's just a matter of efficiency. You should get more or less to the same point. So you learn the parameters from the data, exactly like AlphaFold is doing to learn the folding from folded structures. So that is the true meaning of learning. It's not fitting, it's learning. So we have an idea of how to do the physical simulations, but we still need a method to extract these chromatin structural types, the chemical propensities that create the phase separation, that in turn create the compartments. At that time, this problem was solved with this method which we call megabase, and this is an acronym that I'm particularly proud of because it wasn't easy. And this is the way it works. You look at a log cell, and at that log cell you can read the compartment. This is from the 2014 Rao et al. Paper. And at the same logos, you can read a bunch of experiments, ChIP-seq experiments. We throw them all in. Everything was on the ECODE database in 2016. We throw them in the database. So at the logos, you actually read a sequence. And that sequence is the chromatin type, the compartment, and a series of experimental readings at the same spot. Of these sequences, you have many because you have many different loci on that chromosome. That creates a database. And now you can choose what you're going to do. You can use TensorFlow and build a neural network. You can build different architecture neural networks. At that time, there was no ChatGPT. TensorFlow was a little bit less developed. And with my background in applied math and physics, I'm a little bit skeptical of using somebody else's code. So what we did is that we wrote our own code. And we use, since you're old people, we wrote an old style of things, which is this style here. This is the Hamiltonian of a spin glass, or somebody called it the Hamiltonian of a Potts model. But it's also the Hamiltonian of one of the earliest neural networks. This is the Hamiltonian of a half-field neural network. At that time, what a neural network meant, it meant to reproduce the behavior of the brain. So it was, I guess now you call it a neuronal network. But anyway, this Hamiltonian encodes correlations and fields. You can turn the crank, do a little bit of adjustments here and there for efficiency. And what that allows you to do is that allows you to predict the type given the reading of the experiments. After learning all the parameters in this model, then it converts epigenetics into one single variable, which is the type. What is the type? It's the chemical propensity associated to a compartment. They're not the same thing, as we were discussed a couple of days ago. Because, you know, sometimes you might have a locus that wants to be active, that is an active locus, is marked as an active locus, but since it's tied to inactive loci, it's going to be brought to an inactive space. So compartments and chemical propensities, they're not exactly the same thing. They're very much correlated, but they're not exactly the same thing. So we have a whole pipeline. We're going to go and encode and get the chip-seq. Then we're going to run megabase and get the compartment annotation. Then we're going to get the compartment annotation and run microm and get an ensemble structure. Typical ensembles, couple 100,000 structures. What you're going to do? You're going to make in silico high C to see if it works. This is a figure from 2017. It worked perfectly. Look at the compartments. The compartments perfectly reproduced. Here you have experiment, and here you have the simulation. Very well reproduced compartments. How does the scaling with the contact probability goes? Pretty much perfect. Almost indistinguishable between two biological replicates or two experiments done in two different labs. So we're very happy with that. Of course, there's a question, you know, high C is only tells you some things, so it doesn't tell you other things. For example, high C doesn't tell you about distances. And we wanted to check that, so we looked at fish data. This fish data was made to highlight compartmentalization with probes in ABA or BAB. And what this fish data, which is from 2009 in Eretz Lieberman paper, what this fish data shows is that, you see here this red is the one between L1, is the distance between L1 and L3. Now L1 and L3 are pretty far away. And this green is the distance between an L1 and L2, and L1 and L2 are pretty close. Now, if this was a normal polymer, the closer you are along the polymer, the closer you are in 3D space. So the fact that here they're about the same in the experiment, that's a violation of a normal polymer behavior. What does the theory predict? A violation of the polymer behavior. They're about the same. Also, the distance is pretty good. See, keep in mind, here we're predicting distances in micron straight from chip-seq, which is chemistry. Okay, so this violation of polymer behavior, this other situation, stronger violation of polymer behavior. What does the theory predict? Weak violation of the polymer behavior, strong violation of polymer behavior. So this was very, very strong evidence that we were doing something correct. Then we read the paper from Alexandra, and we started being interested a little bit in dynamics, and we figured out, is the dynamics reasonable? And the answer is yes, the dynamics is reasonable. The dynamics is known to be subdiffusive, is known to be viscoelastic, and is known to be spatially coherent. What does the theory predict? Viscoelasticity, coherence, and subdiffusivity with a large variability among the loci, like some of you have mentioned in the past couple of days. So, we're pretty happy. We made this figure. This is a figure from 2016 that has become quite popular nowadays in the field of condensates. And what this figure wanted to highlight a few things. Wanted to highlight the existence of phase separation and the fact that the phase separation is not complete phase separation, but it is micro phase separation with small droplets that splits and fuse. So I wanted to highlight the dynamical nature of this microphase separation. I wanted to highlight the fact that the microphase separation is operated by these little green things, which are proteins. And he wanted to highlight the thing that that microphase separation, which is operated by the proteins, is actually regulated by the chromatin marker, by the epigenetic markers along the chromatin. And he wanted to highlight the fact that coming from our theory, we couldn't really identify a single structure in these chromosomes, which at that time was sort of an open question. We never found a single structure for chromosome. You cannot say this chromosome has this structure. We always had some rather fluid ensembles. And we say that, and some people disagreed, and
now we know that that's the case, and we know that mostly from single cells experiments like DNA tracing. So we started looking at DNA tracing. This is work from Ryan Chang, and so here you have the data from Bin to Jean lab. You have radius of gyration of these chromosomes, prediction from Migrome. Remember, Migrome has been published a couple of years before this. So this is straight prediction. And the size of the chromosomes is about right. Then you can do hierarchical clustering of the structures, the plots, the graphs, they look pretty similar. So we're very happy with that. And, maybe it's an accidental finding, but what we found a certain amount of dumbbell structures that open and close. This both in the simulation, but also in the data. So this is sort of like, we can consider it a fact, a partial fact, because the sampling is not perfect, and of course there's not many chromosomes, but in the data and in the simulation you have dumbbell structures, and at least for these regions that Jean lab imaged, what you have is that the dumbbell is kind of like empty chromatin, and then you have that the genes are all in the linker in the middle. Perhaps it's accidental, perhaps it's not, but it was an interesting finding. Curious, at least for us. Then we wanted to see if we could predict many cell lines. Now, this is a very different problem from what Jeff introduced in the past couple of days, because he's going from DNA to structure, and he has a certain amount of success in that at a certain scale. Now, this is larger scale, and these are 3 human cell lines, so the DNA is the same. And you can see the structure is different, right? You have this compartment switch, you can see here, and you can see here it's completely different, and here is also different. So these are 3 different cell lines, 3 human cell lines, different structures. What you have on top is still, what you have on bottom is still experiment. What you have on top is the Migrome prediction, and you can see that, we predict what is being observed. So this is very, very, very reassuring. And at this point, we have a certain amount of confidence in that, and this has been sort of like engineered, and we're now working with, this is work from Antonio Oliveira and Rahini, who are here in the audience, and we're working with the ENCODE project, and ENCODE, they're doing a bunch of experiments on a lot of different human tissues, cells from human tissues, and we're doing the prediction of the structures, and we're putting the structures on a public database. You can go, take the structures. Again, each ensemble is a couple 100,000 structures for a specific chromosome of a specific tissue. And you can do what you want. And you can tell us where we did wrong, and we'd be happy to know where are the errors. So this is sort of like all the history. It's now at the engineering level. What have we learned? So early on, immediately in 2016, one thing we learned is that the major energy driver of this organization is actually the collapse of inactive chromatin. So the interactions between the B compartments are stronger. And they tend to go to the core of chromosomal territories. So a byproduct of the inactive regions to go at the core of chromosomal territories is that the active regions of chromosomes, they come at the surface of chromosomal territories. This is a thing that we have observed in the first paper in 2016, and has been confirmed by Xiao Zhang with imaging in 2020. So the majority of the trans interactions among chromosomes, they happen to be active chromatin, because active chromatin, of course, needs to communicate, but also because active chromatin happen to be at the surface of chromosomal territories, facilitating communication between chromosomes. So that's, again, our paper with Ryan Chang. And I think that we discovered, again, straight from the data, was that this ideal chromosome function, which had a functional form, but it didn't have a specific shape, that functional form leads to lengthwise compaction. That is in the data. That is in the high C. It's a fact. And that, of course, agrees with a lot of other things, but one thing was immediately clear from the beginning is that this lengthwise compaction is very much related to the formation of chromosomal territories. So this is an ugly picture that I made in 2015, and you can see that there's existence of chromosomal territories. That's due to the activity of lengthwise compaction, to the motors creating lengthwise compaction. And these territories, you hear a little bit more from Vinícius in the next talk, but these chromosomes, these territories, they show up as these, squares of preferential interactions in the Heisey maps, and they show up as sort of like non-overlapping regions of space occupied by single chromosomes in the famous Kramer and Kramer picture. One thing which wasn't trivial or obvious for us at that point is that this lengthwise compaction is also very much related to the amount of knots there are in a chromosome. Now, long polymers, they tend to form knots. That's what they do. And if you were to simulate, now you need to have a way to quantify the amount of knots, and these things are called knots invariants, and there's many knots invariants. One is the Alexander polynomial, which is rather complicated, even though popular, and another one is the minimum rope length, which is somewhat popular, a lot easier to understand. The complexity of a knot can be quantified by the length of the rope that you need to make that knot, the minimal length that you need for a rope to make that knot. If you have very complex knot, you're gonna need a lot of rope. And if you have very simple knot, you're gonna have very little rope. Now, if I had a normal polymer, how much rope would I need? What would be the amount of knotting in that polymer? This one, 200, whatever units you're using. Of course, it's distribution because there's many different configurations in that structure, right? So you're not going to have one value. You're going to have 100,000 numbers, and then you can make a distribution. And then you can turn on the lengthwise compaction. What that happen? You go here. You remove the knots. This is like a aha moment for us because it was a change of paradigm. So for a long time, again, we know that an equilibrium polymer is knotted. We know that for statistical mechanics. That's how an equilibrium polymer looks like. And then there's this theory from Grossberg, which has been a very important theory, very seminal paper, and that says that there shouldn't be knots in chromosomes. So the theory is that how can you have chromosomes that are knots? Well, you take a polymer, you crumple it quickly, and then you don't let the chain pass, and the polymer, because it was unknotted, stays unknotted. So the key here is that it's a non-equilibrium theory. It's the process that doesn't allow the knots to be formed. And the byproduct of that is that you have structures that look like this. They look like fractal, and don't attack me in the fractal. Everybody has a different idea of what fractal really means, but they do look kind of fractal. And so that's a byproduct of the kinetic process, of the crumpling, and of the fact that it's non-equilibrium. Here, our view was very much confirming that the structures that were rather fractal looking. And, but the process is completely different. We have an equilibrium globule, and this equilibrium, it's an equilibrium of a lengthwise compacted polymer. We know that the lengthwise compaction is non-equilibrium, but you can sort of make it into an equilibrium theory. And then you have this polymer that is essentially a self-unknotting polymer. It's not free. It allows chain crossing. Yes, thank you. So it allows chain crossing and removes the knots going from here to here, actively. So now I have the last 5 minutes. I'm going to fly through the more modern stuff. So this is work collaboration, CDBP, National Cancer Institute in the Netherlands, and Eden Lab. And what they did is that they did Heisey on a bunch of different organisms. And these organisms, you look at their structure, you expect some territorialized structures, you find a mess. And you scratch your head. It's like, okay, what's going on here? Of course, this is 24 organisms, 750 million years of evolution.
Why is it happening? They are all different length of the chromosomes, they're all different number of chromosomes, they're all different structures, and you understand nothing. Now what Olga did here is to renormalize everything, to have 2 chromosomes of the same length. And this is what you see, which is very interesting because you start to see patterns. You see that some organisms, they have territories. And you see that some organisms, they don't have territories, at least in the Heisey maps. And sometimes the organisms that they don't have territories, they have this secondary diagonal. And often the organisms that don't have territories, they have this big, strong interactions between telomers and telomers, or centromeres and centromeres, or centromeres and telomers. So what you really see in all these 24 structures is you either have territories, or you have a combination of the other things. And you know, the secondary diagonal is really a fallback, is the 2 arms of the chromosomes come together. So these 4 features you see in all this variability. And the 4 features, as I was mentioning, they're not uncorrelated. So you either have the territories or a combination of the other features. And so what they did is that they called it type 1 and type 2 architecture. And then you look at the products that are there, this type 1 and type 2 architecture, they appear to correlate with the presence or not of all the subunits of condensin 2. So sometimes you lose 1 or more of the subunits, and there's a correlation between the type of architecture and the existence of a functioning condensin 2 complex. So this is where the theory comes in, because I know what's happening here, and I have an idea because I have this theory, and this theory has phase separation, and it has lengthwise compaction, and I know that the lengthwise compaction is due to the motors. So what happens when I lose a motor? I lose the function of a motor. Well, the lengthwise compaction should be going down, right? So that's sort of like, that was the prediction of the theory. So if I do an experiment, I should have a wild type with territories with a healthy amount of lengthwise compaction, and then I remove some lengthwise compaction, and perhaps I'm gonna see the clustering of centromeres and telomers happening, and perhaps I'm gonna see the territories weakening, and that's exactly what happened in the simulation. So we had a theory of why there are these 2 types of structure. The 2 types of structures that are created by the competition of 2 molecular mechanisms, which is one is lengthwise compaction and the other is phase separation, and they fight each other. And by fighting each other, you can fall one way. Everything clusters, heterochromatin gets together, the telomers and centromeres get together because they want to be together because they're phase separating, or the mothers, they don't let that happen. And the moment you stop the models from letting that happen, the natural tendency happens. And so this was a prediction, and the experiments proved exactly that. So they knock out condensin 2, and they see that you go from this phenotype that has strong territories to this phenotype that has weaker territories and clustering of centromeres and telomers. So you can play this game more, and this is, by the way, this is work from Sumita Brahmachari that is in the room, Olga Dudchenko is in the room, Claire Hohenkamp who's in the Netherlands, and then Sumidaba went ahead, did more work, and you can, by modulating the activity of the cohesin and the condensin, you can create a whole zoo of different structures for chromosomes that go from completely globular, collapsed, from random walk, but also to stuff that looks like ropes and strings, with the ropes probably looking a little bit like the vermicelli that have been mentioned. But being Italian, I hate the name vermicelli because it's, but so we call it rope-like. So this is work from Sumidaba, and Sumidaba also looked at sort of this molecular mechanism fighting each other, right? So you have that the phase separation generate the telomere compartments, but the chromosomal territories, they fight the telomere compartments, and then you can add the lamina, and you can create this diagram in which you see how the things interact and fight each other. So I'm done with this. Just take-home message. Take-home message is that they're all things you know, essentially. Chromosomes are fluid, yet organized. Genome organization results mainly from phase separation motor activity, and epigenetics control genome architecture along differentiation by means of controlling the phase separation. And, and this sort of like this is a high-level theory, okay? It's not meant to reproduce the detail. But this high-level theory of two molecular mechanisms, phase separation, lengthwise compaction, really explain a lot of the architecture of different organisms along evolution. So we have sort of like a general understanding of how things happen at chromosomal scale. That's an important caveat. Here we're always talking about chromosomal scale. Lower scales? I'm not sure. So acknowledgement, this of course is not work from me, this is work from a large number of people, the Center for the Biology of Physics, my group here in Northeastern, CP, Jose, Eretz sitting in the room, collaborators in the Netherlands, all the fantastic team of people working on all the simulations, Antonio, Mateus, Vinícius, some of my colleagues that were at CDBP are now around the world. The funding is, of course, NSF. My lab is also funded by NIH. And important to mention the Massachusetts company facilities and AMD for donating a large number of GPUs. So we are always hiring. Typically, I show these things and we're not here, but you can look outside the window. Our office is down the street there. And thank you very much.
Questions?
Thank you. This is really fantastic. I'm curious whether you have compared your structures with the GAM profiles in comparison to Hi-C. So in the latest multiplex GAM, we learn about Hi-C being poor at capturing these complex contacts. There's some bias for contacts that are more simple, placed within heterochromatin. And I just wonder whether you have looked into whether you catch those more open chromatin involving many contacts.
Yes, that's a good point. We have looked at GAM in a couple of different occasions. I personally looked at GAM a few years ago, and I have concluded that the resolution was not enough for me to draw conclusions. I now have people in my lab that are currently looking at GAM with the help of Anna Pombo.
All right.
The answer is in the works.
And one more, if I may. So do your structures also recapitulate properly differential density of chromatin across nucleus?
So, well, differential density is a very difficult question. So we looked at density. We looked at density again with a couple of different models at different resolutions. There's a large variability of opinions in the experimental world, so it's very difficult to know who we should listen to. In terms of differential, I'm not sure there's data that I would, confidently rely on. But we are looking at, particularly we are looked at different densities using tomography, electron tomography from, what's her name, Oshia.
Thank you.
So I'm wondering about when you're modeling the polymers, how do you add in the fact that, at least in dividing cells, every 15 kb you have a topo 2, so the polymers just are invisible to each other. And if you think about that, then it might be interesting to compare proliferating with non-proliferating cells, because in fact, non-proliferating cells have much, much, much less topo 2. So you could actually, there are natural experiments where you can vary the amount of this breathing through the polymers.
Yes, that's a great question. So these are part of the things that we took care, but there's never time to explain. I'm glad you asked. So when I say we start with a polymer, well, there's many different polymers you can use to start from. And the polymers, they can be chain crossing, they can allow chain crossing or not allow chain crossing. Neither one is good, because topo has some activity, it allows the chain crossing, but only every once in a while. And you can, so what we have that we allow chain crossing every once in a while. About every 1 over 1,000, 1 times every 1,000 tries. In this specific model, we allow chain crossing. Of course, that's going to depend on the activity of topoisomerase in that specific cell type, like you said. So we do take care of that, and the dynamics will depend on that strongly. And there's some work in the progress looking at different more proliferated cell types and less proliferated cell types, but at the moment I don't have precise answers. So we allow occasional chain crossing, differential chain crossing different cell types. We're looking into, I don't have an answer yet.
So you just said that if a lineage that would be extremely interesting and is very accessible to look at is chicken erythrocytes. Because we showed that at 10 days of development of chicken erythrocytes, you lose TOPO2. It becomes essentially undetectable. But in that same lineage before that, there's plenty. So you could have the same lineage with and without the ability to do the chain crossing.
Thank you for the comment. This is, I didn't know, and
I can't them from eggs.
Yeah, it's fantastic. I mean, well, actually, we looked into that. Thank you. That's a very good point.
That was really beautiful. So I have this sort of like, I have the same question as Bill, but I was thinking of people that are treated with cisplatin or any topo inhibitor, etoposide, for cancer treatment. So if Bill's model is correct, you might think about looking at cancer cells that have been treated with etoposide, and you might actually expect the high-C map to look fundamentally different if it relies on topo2 for disentanglement. And then the second question was, if you look at the same process over developmental time, there's a very strong correlation with linker histone changing nucleosome repeat length, right? So your assumptions in all these models for everybody here is the spacing is identical in all these different cell types. So that's actually not factually true. So I'm wondering, like, if you just play with the bead distance, like, each bead, and you change linker length from 195 base pairs to like 205, which is what you see like in some neurons, does the high-C map change, or does it not care? Thanks.
It's a fantastic, they're all fantastic questions. This audience is so ahead of the curve that you are asking me all the things we're doing right now and we haven't finished. So we published a paper about, so this model is 50 kilobase resolution, so we cannot do with this model what you're asking, which is spacing. But we know that's a limitation. So we now have better models, but with good DNA mechanics, and we can change the spacing. My postdoc, Gaurav Bajpai, is in the room, is working on that. We have a paper published on PLOS Bio on that. So we're looking at that. And in particular with Herbie and Sumitaba and Jose, we talk a lot about topoisomerase. I mean, this is clearly the elephant in the room. There's going to be many elephants in the room today, but so that's an important thing. We're looking to that. It's not an easy. They're not easy, the things you're asking. So there requires a certain amount of developments of theory, and we're getting there.
Next year, this time next year, we'll wait for the answer.
Yes. This time next year in Corsica.
So, Michele, here.
Yes.
Very nice. Good to hear from the horse's mouth, right, as they say. So, great. So, a couple of questions to clarify your approach. So, the activity is taken care of here through interactions, that gamma of D, that sort of thing, right?
Yes.
And so, so the way I understood it, the temperature is the same across all the, or what really is the temperature? And, activity can also be taken care of through temperature, like, topoisomerases, etc., is pumping in energy, right? So I was just wondering what you think about taking care of that through interactions versus other means.
Yet another fantastic question. Thank you. So this is a model which is really, it has the shape of standard statistical mechanics, but it's really information theoretic model, right? We are learning.
It is what?
An information theoretic model. We are learning from the data, so we're inverting the data. So in that sense, there is a temperature, but that temperature is a fictitious temperature, which is characterizing the activities, as you said. So that temperature here is 1. And temperature 1 means room temperature, where they did the experiment. This stuff is sort of calibrated to represent the activity that was observed in the experiment. But we can still change it, right, as you said. And in fact, Vinícius, in about a minute, will show you what happened when you remove the activity by lowering the temperature. So that's sort of like a standard approach in condensed matter, and we do look at these things too. I'm gonna let Vinícius, I'm not gonna steal the thunder from Vinícius.
Yeah. So, and you said this is more at the level of the chromosome. So what is the sort of length scale? Chromosome, we know what the length scale is, but actually it's below chromosomes, right? The models.
So the model, each bead of this polymer model represents 50 kilobases of DNA. So a chromosome is a few, like let's say 100 megabases, so there's a few thousand 50 kilobase beads in these chromosomes. And that's sort of like the scale at which we started 10 years ago. Now we have models which are nucleosomal scale, DNA nucleotide scale, and there's a few variants of that. And also we're scaling these models from single chromosome to whole genomes, which means multiplying everything by 46. And there's problems in converging and other stuff. But so it's not easy, but we're going both up toward the whole nucleus and down toward the nucleosomes, because we know these are important things.
Which related to Olga's question, can you predict that?
I think it might be off, but...
Sorry. Related to Magda's question, I was wondering if you can predict LADs.
So, fantastic question. So, we know LADs are important, and Sumidaba has put in this interaction in the simulation. Can I predict LADs? Not at the moment, because megabase was meant to predict chromosomal compartments and our LADs. With Ryan Cheng, we are working toward generalizing megabase to predict other types of compartments in the work.
I have a question, which is that you have this effective description of the polymer structure of the genome. And so there's some evidence that you can reproduce dynamics in some cases. I wonder if you can just say a little bit.
Sorry.
There's some evidence that you showed that you can produce the dynamics in some cases.
Yes.
I was wondering if you could talk a little bit about sort of where do we expect this kind of model to actually reliably reproduce dynamics? Like what time scales, what length scales?
So this is it's a very good question. It's a little bit complicated. So I had no expectation that this model would reproduce the dynamics, because the model is really based on thermodynamics. High C is thermodynamics. It doesn't know anything about dynamics. So I had very little expectation. Actually, in that sense, the model has outperformed with respect to what it was reasonable to ask. So I don't think this model is a good model for dynamics. So straight to say, I think we reproduce some of these features, but the features are fairly general. Other models that reproduce them as well. So that was sort of like, it's a reasonable dynamic. It's not a good one. So now back to your question, it's like, what should we do to get good dynamics? That's a hard question, because that involves figuring out what is the frequency of chain crossing, which is due to topoisomerase. It's a big problem. And there's a problem of how you're going to do that, right? You know, what kind of polymer you're going to use. And in most of these polymers, actually DNA is not there, right? Even all these higher level models, which would have nucleosomes, they have the nucleosome, but then they're connected by springs. So the short, so the answer to that is, I'm not sure. We're trying with higher resolution model with nucleosomes, but it's not easy. And the second and the last difficulty is that I am not sure what number I should reproduce, because I see numbers all over the place from experiments. So we're working on that. But what is the dynamics? Do you have do you have an idea what dynamics? What is the dynamics of a chromosome?
I think other people in the room know better than I do.
So it's not, you know, I'm open to suggestion in this. And then we'll try to do our best to explain that.
Okay, well, if there's no further questions, let's thank Michele again.
So hi, I'm Vini from José Onoshik Lab. And before this actually start, I would like to thank the organizers for the invitation and also for putting together this nice event. And I also want to thank Michele that he basically they give a great overview of many of those ideas that we'll be discussing here. But Michele covered, sorry Michele, a lot of the parts on the human in interface, and our idea now it is how can we do one step further and explore how we can see the chromosomes change shapes not only in the interface but across the cell cycle as well, and also model different organisms when the chromosome is changing from interface. Our first question, as I say, we are really interested in observing that be actually the experimental data that Bill presented 2 days ago. Jober Decker also showed us, they covered a lot of the details on how they have this very nice synchronized high-C data in different time points of the cell cycle, which was very interesting for us, because the data that we had for human that Michele showed, we covered a lot of the G2 or 0 minute in there, which is an interface. Then we wanted to explore what is happening during the rest of this transition using those maps. Okay, so that will be our first approach. Just to recap, the model that we will be using, Michele covered it, but I would like to give my view as well. We call the minimal chromatin model. The idea is exactly to be minimal. So instead of putting a lot of the details on things that will be sometimes more harder to comprehend, we try to narrow down in 2 key physical interactions that we call, in orange here, types, and the ideal chromosome. Types will be basically, in a more rough and naive speaking, is if I have my polymer with 2 color beads, how is the interaction between them? What's the flavor of my polymer? Okay, so red would like to stick with red, green would like to stick with green, very simple. And then we have another energy term, which is related to all those different proteins and motors that can be acting in the polymer, as Michele said, will be a lengthwise compaction term that will help us and facilitates to get the correct compaction and the polymer scaling, as you saw. They have been pretty much successful in reproducing human high-C data interface, where you can get information about TEDs and compartments and many more that you already saw. So then this will be basically our 2 tools, experimental high-C data, and then we have the theory in our hands. Let's put them together and start modeling those different time points. As a first result, we took the time, 0 minutes, chicken in interface, and as expected, we produce many of the features that was observed in the human interfaces as well. The structure will be a more or less a globular shape, which is also observed in the human. We have longer range interactions that favor this type of configuration. We observe phase separation, and we can also get the polymer scaling correct. The way that the genome is compact, we also get capturing using this very simple model that, again, just accommodate phase separation and motorization, which is very nice, but so far, not a surprise. Then comes the challenge in how transferable this type of energy function could be for the different time points. And as a second step, we did the modeling for the genome, for the chromosome in the prometaphase. And we got most of the features that they observed in the experimental data as well. So the high C, they show they lost longer range interactions. The chromosome, as you can see, they form the rod-like shape where the ends don't talk much with each other, but they have this cylindrical shape. Although here we only have pairwise interaction, we haven't added any artificial confinement to this polymer. So this comes straight from the mono and pairwise, which is also very interesting that we can capture the two shapes only by internal interactions. And as a consequence, the polymer scaling is also obtaining there very nice, especially when we have the second diagonal, the second band, and then we observe the peak, how the contact probability decay is also there in the model. So that was very nice that we are able to get with a very simple energy function those different times of the cell cycle, and we actually did this for all. And then we can consistently tracking the time evolution on how the structure change and how this is reflected on the high C. So for now, it's fine. We just reproduce what the experimental data have in there. Okay. We wanted to do one step beyond, which it is what it is then the structural implications that they have in such calibrated force field. What else can we learn and extract from this type of model? For doing that, Michele actually answered some of that. The simulations that we run we have some temperature, information temperature, that creates a lot of the thermal noise, which help us to sample the data that we need. The strategy that we're going to imply now, it is, I wanted to look at the minimal energy state. It's a very common strategy, which is we start with that I want to see what's the native state, what's the minimal energy high temperature. For that, and then slowly we cool down the simulation, which means that it gives enough time the polymer to accommodate local interactions, and then if eventually you may have some defects in the polymer, they should diffuse to the end of the chain, and we should expect to see something a little bit more orderly, which may represent the tendency of the force field that we have. Okay, so then we'll be using this quantity strategy to see then the structural implications. Okay, we applied this strategy using the force field for the different time points. And one thing that we can already observe by looking those nice structures, actually I think that's very nice, it is the formation of those helix. We do have some coils and some turns and some helix forming, starting less evident in the interface, but when we reach 15 minutes, it's the prob-meta phase, the helix become very evident. Okay, as you can see, and also in the different layers by the different colors. Okay, so that's one nice observation that comes naturally from the data. So we are not implying any artificial coil in the model. To quantify a little bit how consistent are those helices, or what's the frequency, we calculate a kind of, we say, orientation order parameter to try to map and quantify how significant are those turns and what is the frequency that they do. It may be a little bit harder to see, but they look similar to a radio signal. And if we zoom in, especially on the prob-meta phase, besides those high-frequency turn that is related to those high-frequency helix, we also have a kind of a wave. There's a second wave in there. So what's that's actually those two frequencies mean for us? To try to understand what those frequencies means, we did a Fourier transform in the signal. It's a very common strategy, if I'm able to click on this. Okay. we apply a Fourier transform on that. So the peaks will give us what is the most common frequency that distinguish from the rest when we apply in these structures. And we can see nicely the progress of the enlarging number of the distance. So we start with minor coils around 130, 150 kilobase that span around 450 kilobase until right before the start of the prometaphase, or something between the prometaphase and the prophase, which is very consistent with the extrusion part of the condensing tool, that those loops are increasing size during this phase. But again, we have not explicitly included any of the motors. This is just come from the data. But when we look at the prometaphase, we observe two peaks in the Fourier transform. The one to the right is the high frequency helix, which is something around 100K, which is interesting, auto-consistent with the nested loops when we have then condense 1 also acting for the condensation process. But I would like to point out another interesting aspect, which it is these peaks on the left, which is totally related to the second wave that we have in this our radio signal here. So this peak is telling us that the polymer is organized. Besides the high frequency turns, they also have a low frequency periodicity in the mitotic, the prophase genome, that we can, as Bill commented, that we can call this a sort of a backbone of the chromosome. I think that this was very, very, very interesting that we can get this without explicitly saying that, okay, you should turn or you should coil or you should compact. We also calibrated and tried to observe if any of those turns, if they have any preferential direction to go, if they could be turning for right hand or left hand. So, I know that is maybe a little bit polluted, but the idea is that we observe no preferential chirality in either of the regimes. So our polymer, they can choose either to twist to right or to twist to the left hand. And doing the quenching, I did the coloring here of the structures where those white spots represents some defects in our structure. When I say defects, it is this region did not choose any of those chirality, those turns. Even though the quenching strategy was to go to more ordering, we still have some defects that are present in the polymer. We're still investigating some of those perversions and defects in the polymer. Okay, so as I promised at the beginning, we were going beyond human interface. We covered the chicken, many cell lines. I also want to comment the genomes about our invertebrates as well. Things that we see on the high C, that they do not necessarily form a clear territory when look at the high C. And for that, we choose the data from the mosquito. It's the Aedes aegypti high C maps performed by Aidan Lad and by Olga, that I would like just to highlight a few things on this map that I think it will be interesting for us. So in the intermap, we have one dot here, one enrichment that tells that centromere and centromere they have some some colocalization in 3D space that is captured by the high C. We also have contacts happening between telomeres to telomeres, and I call here a strong diagonal. It's strong when we compare to the human, and then we also have an anti-diagonal formation. So put those ingredients together, it suggests that the mosquito will have a genome architecture or a genome organization as the so-called Rebel-like configuration, where centromeres and telomeres are polarized, forming this hairpin-like structure that is very common observed in plants. So we apply very similar strategy as I comment on the human and on the chicken. That it is, we use experimental high C as input. We learn some of the parameters, and after the optimization, as Michele covered, and then once we have our force field, we applied quenching again on the structures to try to observe structural implications on this force field. And which is also, I think, is also very nice, that we obtain most of our initial assumptions about the high C, where we have telomeres polarized with the centromere, look into the structure, and the colors here go from blue to red, so we also form a nice hairpin-like structure. And from the structure, we can also see that they do have a formation of a high-frequency helixes forming here and there that we can track. Doing a similar analysis, we obtain two major regimes of coils, around 600 kilobases, and a high frequency, and 8 megabases on the lower frequency, which is at some point similar to what we will observe in the pro-metaphase mitotic chromosome, which suggests to us that the mosquito genome, they are organizing the nucleus, what we call a liquid crystal, or it will be partially condensed. And then when we say a liquid crystal, it's mostly because of this competition of, we do have local ordering and formidable helices, but we still have some fluidity between those two arms that allow us to observe some compartments along the anti-diagonal. So I think that's a very, very interesting system. So we decided then to put all those chromosomes together using the calibrated force field in chromosome 1 to have a sense of how the whole genome of the mosquito will look like. So we have two copies of each chromosome, paternal, maternal, and we do observe they keep the polarization of the centromere and telomere. They form this type of elongated configuration, even though it's not clear. It's clear, or we can say it's more or less clear from the high C, that experimental high C, that they don't form in territories, but when we actually look into 3D, they have these elongate configurations. We can say that they don't overlap too much, but it facilitates them to get those intercontact that we observe in the data. I'm going to skip part of our computational development. I just wanted to summarize most of our findings here, that especially for the chicken, we observe the formation of a helix when the chromosomes are in the pro-metaphase, in the mitosis, and with those two characteristics frequencies, which is consistent with the different motors and models that people have been proposing. We also observe no preferential chirality, and we also argue that they may be being organized in a liquid crystalline fashion, where we have, again, local ordering and fluidity to accommodate the compartmentalization and other interactions. And all the tools that we use is now hosted at Nucleome Data Bank, hosted at RISE. So Sumitabo gave a nice workshop on how to use those tools. I invite you, if you have any interest to take a look on that. So with that, I would like to thank all our collaborators that were part of this work, especially Professor José for all the support. Of course, the funding agencies, NSF, and the AMD that provide us with the computational resource. So yeah, I'll be happy to take any questions. Thank you.
Maybe following Bill's suggestion, I'll invite a student or postdoc to ask the first question, please. Somebody must have a question.
Hey, thanks for the nice presentation. I was curious, so I didn't quite understand in the model, which parameters do you adjust when fitting the different cycle stages through the mitosis? Yeah, thank you. Nice question. Okay, so getting back to the model. So the energy function that we use, they are transferable for all the different time points. And the parameter that we need to adjust, it is how strong will be the interactions in those two terms here. This alpha, which it is the flavor between those bits, and the gamma, which it is the lengthwise compaction, how strong they should interact over the genomic distance. So if we go to our results that we observe, the weakening in the compartmentalization do not necessarily means the weakening of the forces, but the difference between we say AA interaction and BB interaction, this gap, they decrease, but the overall energy to have the polar remains more or less the same. So these are the two patterns that we fit.
Hi, over here. Beautiful talk. I just wonder, because looking at these models, we don't see condensation of mitotic chromosomes. They look like having the same volume, which I guess is because the bit size is still the same. But I wonder, in the model, would it make sense to vary the size of the bit depending on the ataxic signal, for instance, to account for higher or lower compaction of a locus, even though I hear that ataxic data that doesn't change that much. I don't change that much in mitosis, but I just wonder whether it would help.
Thank you. Yeah, that's actually a very nice idea that we are basically work on it. So regions that we have a low signal, we keep the distance between the first neighbor more close. Then regions that are open, we expand it a little bit. But I don't have any results on that direction. But we are working on it. That's an interesting idea. Thank you.
Thank you.
Yeah, it's really interesting. I had a couple of questions. First, about ATAC-seq. We just did some analysis of RPE1 ATAC-seq, and it's just incredibly important for gene expression, of course, but don't forget that it's like 0.3% of the genome. It's open. So you're not actually talking about a major effect on the genome. So my question is, and I probably just missed this. So you said you had a low and high frequency that you found with your Fourier analysis, and the high frequency was 100K. What was the low frequency?
Yeah, so let's take the 15 minutes per metaphase. So the low frequencies, it sits something around 2 megabases to 6 megabases, which is more or less in the range of the bump that we have in the contact probability or in the second band from the high C. And then they, of course, they shift during the time progresses.
So something that Yoba and I didn't say in our talks is that the gyre size that we use is about 10 megabases. So it's very, very close to that. And Kimiko has actually measured the gyre size 2 different ways by microscopy, and she also comes up with 10 megabases. So that's really interesting. Then the question, I didn't understand, what's the biological correlate of the quenching experiment? So biologically, because that's pretty critical, because it's the quenching that seems to be producing this helix, right?
Yes.
But we don't I don't understand, or I don't think I understand what's the biology of making the helix. So can you help me out there?
Yes. So two things. So the quenching strategy we use a lot in the property community to see the minimal energy and remove those thermal fluctuations. And in nature, we observe a lot of the tendency of forming helix all the time, right? In proteins, we have the helixes, DNA, we have the helixes here as well. And then we are only seeing the implications of the force field. So we can say that in some sense, this information is already on the data. We are just trying to filterize the thermal fluctuations on the noise that we have in our information temperature. This could be one way to respond to that. And then on the second way to respond, that's more about my opinion. When we look at the high C, for example, all the data is cross-linked, they are basically frozen, which is something similar that we are going to this low temperature. So we may have hundreds of thousands of those quench structures that they may reproduce high C. We actually work on that to see the consistency between the high temperature that we use for the model and the low temperature that we use for analyzing the structures.
Here. So in your Hamiltonian, in your model, there was no liquid crystal type interactions. But in the end, you mentioned liquid crystals. Is it that these beads are aligning that you can assign a director and then infer a liquid crystal type photoparameter?
Yeah, thank you. I should have clarified a little bit better. So the order parameter that we are using is not the numeric that we have the director and to see those ordering, but the order that I'm speaking is about the helical ordering that is invariant along the polymer chain. So that's is where the crystallinity part comes from this local ordering that's translation invariant through the polymer. We don't have the direction or the director vector as you comment. So yeah.
Okay.
Vinny over here. That was a very beautiful, very lucid talk, really gorgeous slides. So I actually downloaded your paper and I haven't read it yet. So I'm going to ask you a dumb question. So you said this in passing, that the coil didn't care if it could go left-handed or right-handed. And that was actually kind of astounding because the origins of supercoiling in bacteria is always left-handed for the most part, right? So why do you think in these structures that you're simulating, the fiber doesn't seem to care? And is there an energy difference between left-handed or right-handed, or can it swap?
Yeah.
And then, sorry, the second question was simply, I was totally fascinated by your centromere clustering. So does it start out at the telomeres? Can you pinpoint the origin of where the fold starts? Like where is it starting? Is it starting at the centromere and then spreading out? Or is it starting at the telomere and then the centromere is the clustering? It would be great to answer both if you can. Thanks.
Thank you. I will answer the question. The second question first. It's because I don't know to give you a proper answer for that. For the first question, I do know that we observe no preferential chirality, more or less as you say, that we have no energy gap or no energy penalty for bending for either right or left-handed. Mostly because we are learning the data from the high C, and then when we have the distance there, it's basically the mirror image. They have the same distance that we do not incorporate this on the model, so we don't actually know if they have a chirality in the polymer. Although there is some evidences that when sister chromatins they are segregating, they do form in the backbone a different chirality in some cases, and then they have the same in the other cases, which is still interesting where we are actually investigating it. Yeah.
Can I just say that in Anton's models, there's also, for our high C data, there's no chirality.
No chirality as well. Okay, good.
That's really cool, but I wonder if you followed up by overlaying, you can't do this for Egyptii, but you could do this for human cells. Dave Levins and Nick Gilbert both published trimethyl sorrel cross-linking maps that give you the supercoiling of given domains. And there's huge domains that are positively supercoiled or negatively supercoiled, right, in plectonemic state. So I'm wondering if you could overlay that data set on your high-C maps, because they're also megabase-sized domains. And that would be fun to see what happens, right?
Just for consistency, Vinícius, it's important to remember that basically he's looking just like Bill is looking on larger scale. These are 50 kilobase bits, and supercoiling is a much smaller scale. So you still may have at the level supercoiling the left hand, but then you're talking about at that scale. We have to sort of integrate all these scales, but just to be clear that we are looking at very different scales here.
Yeah, also to comment. Also to comment, Sumitable is also working the supercoiling model, integrating those aspects that you mentioned, the large scale. Yeah.
I think one more question or two before the break.
Question? Sorry, hi.
Sorry.
So you pick up the two peaks in the Fourier, yeah? But there are some middle, the small one, and do you have any idea what it's suggesting? Especially in the 30 minutes and 60 minutes between the two peaks, there is also some signature.
Yes. So if we have a crystal which will be perfectly ordered, we are supposed to see only the two peaks, but we do have some noise and fluctuation because of those defects, and then some of those frequencies can pop up as well. So it's just...
maybe the remaining or not?
Yeah, it's an interesting idea to check, but thank you.
Oh, thank you for a great talk, and of course I've seen this before. I wanted to make a comment about the supercoiling and top isomerization, because this is a huge problem, and we're all working on this, and it's clear, none of these models can do that. These polymers, they don't supercoil, neither ours or Anton's. They're they don't form plectonyms. And that is, of course, very big failure. And the point is it's not easy to make polymers that make plectonyms. And we're working on this. And possibly when we actually put the chirality of DNA and the cause of the nucleosomes in, it is possible that that's going to break the symmetry one way or another. But none of the currently available model can access this question at the moment.
Can I make a wild suggestion, particularly for the polymer guys who like to think about all of this stuff? Look up a review by a guy named Ueda, UEDA, published, I think it's in the Japanese Journal of Genetics in 1938, which you can get online, all right? And it's how you separate coiled chromatids. And it's chromosome segregation before topoisomerases. And it's amazing how complicated it had to all get. And it's really fun to look at that review.
Thank you.
Well, let's thank Vinícius again. And we will continue again at 10:30 after the break. Thank you. Okay, welcome back. Our next speaker of the session is Vittore Scolari from Institut Curie. And he'll tell us about amine field theory for predicting single polymer collapse induced by neutral Crowders.
Thank you. Thank you all the audience and the organizer for inviting me here. It's a great pleasure. So I will present you a theory talk today. And I would like, in the title, to highlight the word predicting, in the sense that this theory is really a theory that wants to be quantitative, in the sense that we want to learn something about neutral Crowders, and be able, after we learn it, to predict something from an experiment quantitatively. And next, I would like to stress the accent of neutral Crowders. It is something that we didn't hear about today, that is the nucleoplasm will be in eukaryotes. And so nobody's modeling it, and I'm going to talk to you a bit about this and why neutral Crowders, because while this maybe is not important in eukaryotes, I'm not sure. I hope it is. Like, it is a topic that is very important and well studied in other organisms like prokaryotes. So this is the DNA of an E. coli cell after you lyse the cell walls, and you can see that it's quite big. And we know that it is in fact condensed inside the cell in a compartment, so in a specific territory, that is called the nucleoid. And when you lyse the cell, it basically explodes and becomes much bigger and shows all its features. That is, in fact, quite complicated. Like, you can predict, if you want, a radius as a random walk, which is a typical model used for Polymer, but this doesn't make any sense, because, in fact, it has very long correlations, as can be seen here. It has something which resembles loops, something which resembles a core. And so people want to understand that, how the DNA can fit inside this nucleoid. And there are many different elements that one has to consider that are considered very important. One of them is supercoils. Like I've been listening to questions about that. So supercoils are made out of these secondary structures, which are plectonym and toroids. If you combine them together, you obtain a tree. And this tree can be observed by electron micrographs, like the one I showed you before, and this one by the same person. And the people, like a Cozzarelli group, have been going there to measure the length of these plectonyms and estimate their size in kilobases. And what has been shown is that you have a characteristic size which is around 10 kilobases, so it's a domain. And this correlates also to measure of transcription, like a correlation of transcription upon DNA damage. So supercoil is surely an important element in structure in this chromosome inside this nucleoid. Another thing which has been shown to be extremely important, I'm not going to talk to you about this today because it's nucleus-associated proteins, including condensins. So this has been studied in Kosul and Boccar lab in Paris. And it has been shown that, in fact, the structure is not homogeneous. It is heterogeneous. It's structured mostly by transcription. And on top of that, there is compaction, which is also non-homogeneous. And this is structured by condensin and other proteins which interact with it. And so this is something to keep in mind. There are proteins. Another thing which is important, like I would say very important, is it's alive. And this is quite important from the point of view of the dynamics, at least. Like it has been shown that upon ATP depletion, you have a slowdown of the motion of chromosomal lossy by this Weber-Spakos interior 2012. And this happens also upon inhibition of transcription. So it's not very clear yet how it relates to compaction, but there are theories. But it's clear that it has an effect, and the effect is important. And not only on the motion of chromosomal loci, but also on the motion of the cytoplasm, of the called Croders. Like, it's been shown by the Jakob Wagner group that if you put a plasmid, which is called mini, but is not that small, of course, inside the chromosome, inside the cell of E. coli, and you deplete ATP using another kind of treatment, you freeze it, like it doesn't move anymore. So it's been proposed that without ATP, without activities, the nucleoplasm will be a glass-like structure, will be jammed. That will be the physics jargon for it. So it is important. So there has been theories about how this cytoplasm compact the nucleoid inside this compartment that can be seen in an electron micrograph. That is DNA and this is protein. And this one of these theories, the first one I would say, is the Theo-Dyck hypothesis. That he made a theory which is a phase separation, like is very qualitative, I would say, and is similar to what has been discussed in other talk today. And he said that the cause is entropic, in the sense that when you have a big molecule, such as DNA, that is close to other molecules of the same kind, it will let more space to the small molecule to diffuse. As such, you have an osmotic force which creates attraction and compaction. And is the experimental evidence of this? Well, there is experimental evidence in vitro, like from a purified nucleoid from the June lab. This may be the first one, I would say, where they had a nucleoid inside a microfluidic device, and they add and they remove a PEG, So in different cycles, PEG is what is a typical neutral crowding agent because it doesn't interact in any way apart sterical repulsion, like this kind of small liquid molecules. And it's shown that you can compact and expand and contact and expand the nucleoid many times in a way that looks reversible. And this has been also been plotted in a phase diagram that show the DNA volume in function of PEG concentration, and you can see that the DNA volume goes down, so there is phase separation, there is compaction, and it happens at a critical PEG concentration, and this is comparable to what happened inside the cell. And this is also something which arguably makes sense because you want to be able to open this compacted chromosome, so the compaction shouldn't be that much. Open it for transcription, for example. And there is experimental evidence in vivo, and this has been done by Manik Lab. And what they do is that they have a mother machine that is this kind of microfluidic device where you can keep growing cell. And then they change the osmolarity of the media, and by doing that, you shrink like a bag the containment of this nucleus. Like you basically compact it, you increase the concentration of the cytoplasm, and then you look how much the cytoplasm change, how much the nucleoplasm change. And you see, like here is normal growth, then they add this media, and it goes down in very fast, like less than 5 minutes, 1 minute, it's almost immediate. So, and then they can plot this changement in function things like the concentration of this media, of the salt. And they can see that, in fact, the nucleus is compacting more than the nucleoplasm, showing that the effect of a more concentration nucleoplasm results in an even higher effect of compaction in the nucleoid, like where DNA stays. And in all this graph, like you can see that there is a relative concentration of cytoplasma crowder that induced by osmolarity changes like on the x-axis, and then you can see the size of the nucleoid using different measurements, and it goes down. And they did also some simulation, and the simulation are extremely simple, in the sense that they had a confined bed spring model with these beads, the crowders, but it's explicit here, and they have a definition of variables such as the size of a single monomer, which they call A, we call it big D, the size of a crowder, which they call AC, we call it small d, and their density, which they model using their number, and we can model by volume fraction. And when they did this modeling, they realized that they can reproduce compaction of the polymer using crowders, but they can also see that there is something which they call an order parameter, like CAC square or phi over D, concentration of crowder over their size, and this collapse all the curves of the size of the nucleoid. Like if you use this order parameter, they all collapse no matter what is the size of the crowder. So this was surprising. Maybe there is a simple theory that we can make for the effect of collapse by crowding. And that's what we did with a brilliant PhD student now in Institut Curie and Marco Cosentino Gomarsino. Like we applied the general logic. And the general logic is phi over D, where phi is the concentration of crowders, like volume concentration, and D is their size, is an order parameter. Does it mean that it's a simple effective interaction? And the effective interaction is modeled by a square well potential, where there is the hardcore repulsion of the monomer of the polymer and the attraction of the size of the crowders and the depth of attraction. So we want to know the depth of attraction in function of the parameter of phi over D, for instance. And why that? Like, you know, it's all entropy. Why you want attractive potential, which is energy? And the reason is that there is a debate. It has been done already, in fact. And first of all, it was done by Marinduzzo and Cook, where they say, okay, we can model the Kurosawa interaction, which is the standard model for depletion, using an energy, like a depth of this. And this depth have this formula, which, by the way, have phi over d here, but is valid rigorously only for very small Crowders, because it's an approximation for small Crowder size. Then there was other theories, like some of them that used many more parameters and were able to fit some simulations. And some of them used another order parameter. I have to say this was derived in another context, so it might be valid in another context. This was for proteins. But they say it's not phi over d, but it's phi R0, where is the size of the polymer over d. So it was a bit more in depth. And then there was the theory of Genome, that was the group that was already published, that was saying, okay, there is an order parameter that is phi over d. In fact, it's big D, that is the size of the monomer, phi over d. And this is compatible with what has been studied by Marenduzzo. But this is not true, because this is valid for small Crowders. What is true is that they show that this collapse simulation is exactly like the group of Manik did. So the question is this H parameter a good order parameter for effect of Crowder on a single polymer? Well, if you look at the size of a single polymer without confinement, in this case, in function of this parameter, and you do different size of the Crowders, they all collapse. Now, we took these simulations result and we combined them with another simulation that is the one that I mentioned here. And the only difference in this simulation is the size of the polymer. Now, if you plot two simulations together for the H parameter, you see that it doesn't collapse anymore. So most probably the H parameter is valid, but only for a size of a single polymer. That was maybe an idea. So let's get, and this makes sense, because when you look at the swelling ratio or the size of the polymer, this is an extensive quantity, it's something that should depend on n in a way that is also very complex and depends on the different phases of the polymer. So what we moved is to an intensive parameter, and this is what is the virial coefficient. And to extract the virial coefficients from the swelling ratios of polymer, we use the Dudchenko theory, which is the equation of state of real polymer. Like there are real gases, there are real polymers. It is in fact an interpolation formula that fits at the same time the random walk, when you have just a polymer which doesn't have any energetic interaction, with the self-folding work when you have very repulsive interaction, and the collapsed polymer when you have a very collapsing interaction. So all these three regimes are interpolated by this formula, and when we have the swelling parameter, we can get the second order virial coefficient, which is an intensive quantity. And when we plot the second order virial coefficient, which by the way can also be measured experimentally by correlation function, and the H parameter, we have a much better match of all the data of the old simulation that we considered. What we see is that by increasing the concentration of Crowder, or in any case the H parameter, you have a decrease of the virial coefficient, so a decrease of a repulsive interaction, which become at some point attractive, and at some point you have a plateau. So at some point, the polymer is so collapsed that it becomes inelastic to this parameter, to the increase of crowding. So given this simple element, we try to derive the potential, like alla Marenduzzo, so this square well potential and the depth of the potential. We can derive by connecting the prediction for the second order virial coefficient to the plateau size, the plateau in energy, not in virial coefficient, but in energy, and in change for different monomer size. And then we can see that, in fact, a straight line, like a linear approximation, can connect the zero energy to the plateau position. And this is enough to generate, to make a theory, which in fact have a different scaling from the Kurosawa theory that was predicted by Marenduzzo, and is valid for the size of Crowders, where D is more or less equal to big D. So the size of the monomer is the same order of magnitude as the size of the Crowders. Pay attention that this is important because we can do simulation only in this regime. We cannot have small Crowder because there you will need so many of them that it's impossible to do simulation. And in any case, in the nucleoplasma, probably they are not so different, the size. Now, when we do that, we can even improve over the theory for the normalized virial coefficient in function of the H parameter, in the sense that this fits better the data than that. And it shows that, in fact, this is not an ordered parameter, but is a valid approximation for the monomer size close to the Crowder size. And when you go to smaller Crowder size, you will have deviation from the linear prediction. But still, this critical value when it becomes inelastic is the same. Now, we didn't have simulation for larger Crowders. That's quite easy to do. So we contacted some theorist simulators, and we say, okay, why don't we do larger Crowder? Because it's quite interesting. You know, you have this plateau, and this plateau depends on phi, that is the volume fraction of Crowders, and this is limited to 1. So at some point, you can predict you will not have any collapse, because you cannot crowd even more than 1. In fact, there is something which happened at value of density close to 0.6. That is the jamming of the cytoplasm, the jamming of the Crowders. And this prevents the collapse of the single polymer. So this is something to take into consideration and predict a phase diagram that is a bit more interesting. In the sense you have the swollen polymer, you have the collapsed polymer for small volume fraction of Crowders, but when you go to bigger volume fraction of Crowders, the Crowder will be jammed, so you will not have this transition, and something else will be interesting. So there are many things coming from the introduction about what is happening inside the E. coli cell to the reality of the fact that we model a homopolymer without any confinement, without any activity, without any nucleus-associated proteins. Crowders, they were one size. In fact, they are polydispersed inside the cytoplasm. And consider that when they're polydispersed, jamming, which is the last thing that I discussed to you, becomes a scale-dependent problem. Like you will have jamming for very big molecules while they are still free to move the small molecules. Then what about proteins? This is something else that has not been studied by other kind of community, but crowding is very important. What about supercoiling? With the first slide. What about the fact that polymer is circular? Confinement. What about DNA? Because we use this kind of beads which are very round, like these are bead spring model. DNA is not like that. And maybe what about chromatin? And let's focus on these three ones, because this is what we already worked on with success. So what about, the thing is that for a circular polymer and for confinement, there are variations of the Dudchenko theory. Some of them consider the fact that you have different bobs where this is valid and they are aligned all along the cell, when you have confinement on an asymmetric confinement, yes. And some of them consider the fact that you have a ring polymer by keeping the same interaction but reducing the size of the polymer. You can see that more or less the theory fits still all these conditions. Now, what about DNA? And for this, it's interesting that some people working on DNA in vitro, knowing our theory, they thought about doing some experiments. And in their experiments, they can release the tension, and you expect that when you release the tension, if you have Crowder, you will get to smaller distance between the two beads of this optical tweezer than when you don't have any Crowders. So for DNA, you have another important variable, which is the Kuhn length. So DNA is stiff polymer, and this needs to be taken in consideration as an extra scale. And then when you have a Kuhn length, because of the liquid crystalline structure of anything which have persistence, you will have the tendency, in any case, these we don't know, but we know that the interaction will depend on how they are aligned. And if they're perpendicular, you will have very few interaction. when they're parallel, you have much more interaction. So we have taken some approximation to consider this extra degree of freedom and calculating the viral coefficient integrating over the angle of all these Kuhn segments. Like this is quite a hard things to do, but it's algebra, and the PhD student did it very well, also inspiring from literature. And then you have to consider that you have pooling. And when you have pooling, you have three different things that can happen. One is the globule, that's the theory we were predicting before when you have crowders. But then, since you're pooling, you can get the tadpole, and you can get the extended regime. And any of these can be predominant in function of the tension that you put. Again, this we can rely on literature because Alperin and Zulina derived a very excellent theory for these three different elements that consider of attractive interaction that we derived before for the crowders, and we can predict in which regime we are and fit the data. And this, in fact, works surprisingly well. At least for very preliminary and published data, we can see that using 0 parameter, our theory fit their data on pooling. So we think that we are on the right track for DNA, at least. Also, we fit other experimental literature where we have different kind of pooling, and still we are consistent with the result. And since I don't have time, maybe I skip over this. And I go to the take-home message. So we have a non-universal theory that is valid for the bit-spring polymer, quantitative prediction, and this can be extended to DNA. And our extension shows that, in fact, the choice of polymer is extremely important. Like, not all the polymer behave the same. And for DNA, we need an extension that considers this extra length scale. To do, there is all this thing. There is also maybe in vivo chromatin, which we could like pooling this pooling experiment inside the cell. It's something that I find extremely cool that is done in Coulomb Lab. And with this, I thanks for your attention, and I welcome any question. I thanks my group, my institute, my units, and all the collaboration that contributed to this talk.
Questions?
How do we separate ATP effects from osmotic effects, and how basically, how synergistic are these effects? The experiments just look one or the other, but when you look at the model, I was a little bit confused because sometimes you could deal with the concentrations of ATP or the concentrations of Crowders. You want to comment on that?
Yes, it's an interesting question. Like, one thing that one could think immediately is, okay, let's raise temperature, because ATP is raising temperature. This is purely entropic theory, so that won't work. I think we need to think about some specific fluidifying activities of the cytoplasm that doesn't correspond to raising temperature. Like, I can imagine some kind of directed motion, maybe another length scale emerging. Yes?
I don't need a microphone. It was really cool talk. I'm curious, because you're at the nuclear dynamics unit, are you considering using Antoine Coulomb's
magnetic tweezer system where you can do the in vivo magnetic pulling in bacteria, where you can vary the DNA sequence, because obviously DNA stacking, base stacking makes a big difference for this kind of extension model that you have. So if you're like very GC rich, you have alternative DNA structures like non-B form, it's going to fundamentally change your modeling. Are you able to maybe test this experimentally?
It's a really good question. So I think that these can be tested. We are close to testing it in bigger cell, like with big nucleus, because we can pull there. Of course, it needs development, it needs extension to the model, because you have to consider the viscosity. We are pulling only one part of the DNA, not two. But yes, that's promising. I think that we can go in that direction. For bacteria, I think it's going to be harder, but it would be extremely interesting in somebody, typically the locus we are pulling is 1 megabasis. This is already 1/4 of the bacterial genome. That's the difficulty I find, to find enough magnetic force. Yeah.
I was curious, you might have explained this a bit quickly, but you said the square well works well for this swelling of the polymer. What about the kinetics? Is it also recapitulate the kinetics? Can it be described as effective square well, or is it slightly different?
I think that the square well is an approximation that makes calculation more feasible. Like maybe there are also other potential that can be considered. What is important for the square well is that you can modulate the size of the interaction, which is something you cannot do for the linear joint potential, but there are other potential that you can consider. For the dynamics, I think that it's not going to alter particularly the dynamics. But I cannot show you a result. We didn't test that.
So I'll ask a question. So the jamming phenomenon that you showed or briefly mentioned was kind of interesting. And I guess I can understand that it's hard to expel the larger particles from the interior of the chromosome. So what do you expect to happen with the single molecule DNA if you were to go to jamming conditions?
So the difficulty is to prepare the experiment in the jamming condition, in the sense that if this is jammed, it's a different phase, like it's a glass, if you want. So I don't know how you could do that. Yes.
What would it do in simulations?
The simulation is also hard. Like we didn't go to that region, but at least you can. In simulation, there are people that consider the simulation of jammed. You get long range interaction. It's like one molecule which is pushing another, which is pushing another, and this all percolates. This can be done with simulation. There are people that did that. Yeah.
Great.
Can I ask another question?
Yes.
Yeah. You mentioned that PEG expansion is reversible. Is osmotic crowding also reversible?
The experiment of June lab, where they did it in vitro, it was reversible, and yes, it's. I think it's reversible. Yeah.
Okay. Thank you.
Thank you.
Okay, let's thank Vittore again.
Thank you.
Thanks for the introduction. And, with this super somber image, I really, if I could keep a straight face right now, I should really say something along the lines of, I'm going to talk about the elephant in the room. But I'm a bit too wound up and too cold for that. So I'm just going to, without the dramatic pause, say that it's not quite the elephant and not quite in this room. So I'm going to start off talking about, the work that has actually been, in theory, it's supposed to be being published right now. It's 11:00, so, lucky me, no need to add any embargo lingo. Say it again? Oh, yeah, okay. Well, I guess they did a little bit of, even less than I have to do any of that. So this is just out, very, very fresh, and I'm excited to be talking about to you about this work. I'm going to be talking about it actually jointly with Erez, very much along the lines of what, like Michele said, the way that we like to do at CTBP, we're do things from both sides. There's a biological side, there's the physics side. So I'm going to cover what the biological aspects of this work, and Erez is going to continue with the physics. And I suspect that some of the questions that might come up during my portion will probably be answered with Erez's talk, so perhaps we wanted to propose, if everybody's okay with that, with just like the joint Q&A at the end. Okay, well, so, this is an excellent conference, but I don't think that I saw in the agenda or heard any talk from the kind of paleogenomics side, so let me just do the briefest, introduction to help just contextualize the study a little bit. So more or less since like 1984, we knew that fragments of DNA can survive in ancient samples. This has been a very exciting study, and, really, early adopters very, soon shown that, like, kept pushing the boundaries to like how long the DNA can survive. And, in fact, it actually 2 years ago earned those early adopters the Nobel Prize. I'm talking about Svante Paabo. So quite exciting. More and more studies followed. And right now, I believe that the current record holder is 2 million years for the most, ancient DNA fragments that has been found. And the key word in like all of this discussion is that fragments. So that word fragments is quite crucial because everybody in paleogenomics agrees that DNA fragments quickly. So we're talking typically about pieces that are about like 30 million times smaller than the original genomes that were there when the organism died. And furthermore, it's just the fragments. The mental picture that people have is that these fragments are all sort of more or less in a jumble. So they're all mixed up in between themselves and in between the contaminating organisms, really like no remnants of the original structures, as you, for example, can see, in this very, very typical illustration from the Nobel Prize press release. And, that's quite unfortunate because, there's so much we can learn and, thanks to the efforts that have been done including by many people in this room and like how much we can glean from the 3D structure of the genomes. And wouldn't it be exciting if we were in fact able to find some of the organisms, the ancient organisms in which this whole, jumbling would not be necessarily the case. So really, it boiled down to the core question, could the three-dimensional arrangement of DNA survive ever? Is it going to be always, some version of the Nobel Prize picture where everything is jumbled at all scales? Or maybe, despite the fact that the DNA is all fragmented out, maybe under some circumstances you can still imagine that somehow those fragments still remain the original arrangement, and as such, maybe we can come up with a way to query them. It's not like this question didn't, necessarily, just nobody thought about this before, Like probably there was interest in this all the way back in 1984. It was just that in order to query, things in 3D in extinct species, you really wanted to make sure that you had everything working in extant species. And basically, to really start thinking about it, we had to wait until the 3D sequencing really kind of got established and got robust enough to start even thinking about pushing it beyond the tree of life. So basically by 2014, I think we kind of had it reasonably covered, both on the biological side and again, thanks including to a lot of effort at CTBP on the theoretical side, understanding what we're dealing, what the limitations are, that we thought, felt confident enough to basically launch our 9-year fossil hunt. And this was really led by Marcela Sandoval Velasco at the laboratory of Tom Gilbert at University of Copenhagen, as well as Cynthia Perez Estrada at Eris's lab at the Center for Genome Architecture at Baylor College of Medicine, and she's also at CTBP. And it was very, very courageous on their side, on their part, to start this because, again, when we started it was an interesting idea. Wouldn't it be cool if something like that would be found? It wasn't at all obvious that something like that would, in fact, at some point be found. And in fact, they've spent 5 years, pretty much fruitless, sample after sample testing and not really seeing anything that looked plausible 3D genome architecture until finally in 2020 we kind of had something that looked like a signal, and really kind of flagging the special that first case, that special sample should really be credited to Juan Rodriguez in the lab of Martí Renum at University of Barcelona. So they were really the ones who said, whoa, wait, don't move on to the next sample. There's maybe something interesting over here. And really since 2020, this is what we've been doing, kind of characterizing and diving into what we can learn from that ultimately culminated in this study that is coming out, came out. I need to get used to saying that, I guess, just a few minutes ago. Right, so of course, in the 20-minute talk, I won't have time to tell you every single sample that Cynthia and Marcella have tested out, on this 9-year journey, but I will tell you about that one, special, of course, our favorite sample. So where was it found? We found it in a place called Belaya Gora in Siberia. This really coordinated this effort by folks in Yakutsk, Valery Plotnikov and Albert Protopopov, who are our collaborators and co-authors on this manuscript. Just to give you a hint, the romantic feel of how these fossils are found. Here's an actual footage of Lou Vedalin, who was part of an international expedition in 2018, went down a permafrost cave and actually took footage, like this, with I don't know what they're called, head camera, GoPro, this is, I guess maybe the contrast is not really good enough for you to properly appreciate it, but I will show you some still images, so don't despair. And and so really, once we kind of found, once we were able to get that sample out, we were able to understand what it was. It turned out to be a skin from a woolly mammoth, and you could potentially describe the rest of the work that we did as kind of sequential. We just kept zooming in on this sample. So there's the skin, and we, every time what we kept asking is what can we find out if we just look a little bit closer? So when we took it out again from the permafrost cave, what we found immediately is that indeed it was woolly. So that was a relief. You can see the hairs. You can see actually, this is Valery Plotnikov, who I mentioned, and Dan Fisher examining the sample sort of at the field site. So that was exciting and didn't require any special equipment. So, we could glean some structure at the scale of millimeters. But really, to do a little bit more, we had to take it into the lab and sort of, and to actually look at it under the microscope. And so we were lucky to have collaborators for this at Novosibirsk. Елена Кизилова and the folks at Alex Grafodatsky lab really helped out with this. They've done some really, really beautiful images, the histology, among other things, actually characterizing this sample at a smaller scale, among other things, obtaining the first woolly mammoth's hair follicle with hair, still intact inside. So that was very exciting. And even furthermore, as they continued to zoom in, as the histologists can, they actually saw, some of those dimples and bumps that not necessarily tell much to me personally are actually consistent the way that nuclei look. So they're basically very excited that, it seems like there may be remnants of cells and nuclei in this sample. So, okay, great. So we're getting kind of close to some really, really interesting scales. We're basically approaching nuclear, except for right at this point, really histology can't tell you much, and you're really out of your depth as far as the kind of classical toolkit goes in the paleo space. We really needed to start sequencing, but of course, sequencing not in your usual way. So we really needed some sort of a new method, and you guys have heard from several talks, including today, and I'm sure before that as well, about that Hi-C has been really popular and very fruitful in terms of kind of creating 3D genome architecture. So we developed basically a variant of that, and by saying we, I really mean, again, Marcella and Cynthia, who've done a tremendous amount of work for that. So we came up with a protocol. Basically, it's a version of in situ Hi-C that is capable to, it can actually work with paleo DNA, and which tends to be, not only short, that's one thing, but also damaged in all sorts of ways and having been quirky and chemically. So, we basically developed this method, we applied it to the mammoth skin sample, and so what did we find? So in order to kind of understand what we've got here, what we did is we aligned it to the African elephant draft genome assembly. And this is the only thing that was available at the time, sort of, the closest thing. What else could we do? This was the most kind of reasonable thing to do. And immediately we were very, very, this is what we saw. This is what paleo Hi-C data from the sample looked like. And this was very exciting to us because, it's not just some, noise and random jumble, right? There is clearly some structure on this map. And, from our previous work on kind of interpreting 3D for genome assembly, we actually knew pretty well how to read this sort of map. So what we saw here is that, right, there were these squares along the diagonal that basically tell you that, oh, the mammoth had like this sequence, from here to here actually was consecutive in mammoth. And, here you get some sequence that goes from somewhere else in the genome. And, you saw things like this kind of, enrichment far away from the diagonal that tells you that this piece and this piece talk to each other awfully a lot, actually suspiciously a lot in 3D, perhaps, so much so that they probably should be moved and joined together because they're probably neighbors in 1D. So we kind of know exactly what to do with sort of data. And so when we did all of those joins and splits and everything else, what we got is what you're seeing here on the right, which is, is a genome assembly of the woolly mammoth. If you're a really quick counter, you can count that there are 28 squares, which represent 28 mammoth chromosomes. That's the first ancient karyotype of any kind. So we're very excited. This is a first chromosome length genome assembly of an extinct species. And in here, perhaps it's worth pointing out an important point, right? There have been papers that kind of have talked about putting together a mammoth genome. What those really mean is that they sequence those small little bits that are, those jumbled DNA, they align it to the elephant and they annotate the differences. And it is an incredibly powerful approach that yields a lot of biological insights. However, right, that approach does not necessarily tell you that, this 100 of bits and the next 100 of bits with whatever differences there are really nearby. You either have to just take it for granted that, maybe they're in the same order as in the elephant, maybe not. You don't really know. So now we basically resolve that. So we kind of not just get those differences, but also the whole context of the 28 chromosomes. So we're very, very excited about that. And by the by, right, like the fact that you were able to count those 28 chromosomes means that you were looking at chromosome territories. So that allows us to answer, go back to those zooms. And right, we just answered our question about whether we can zoom in further and see chromosome territories. So indeed we do. And, of course, why not just continue keeping going, right? So what about compartments? In human maps, we know how to look for compartments. We zoom in and we look at the plate pattern. So what happens if you zoom in the mammoth? Yeah, well, we also have a plate pattern, maybe not as crisp, but, on the account that, yes, in paleo samples, the amount of data we were able to get is not that large, but it's clearly there. And we can enhance it further by doing like Pearson correlation analysis. We can formalize, who's open, who's closed, the way that we all know to do by using eigenvector analysis. And this looks really, really nice, to the point where we decided, maybe it's even so good that we should compare it to the elephant. So we, tapped into our Zoo collaborator network, procured an Asian elephant skin sample, generated the same kind of map. Here it is on the lower left, the raw data and the Pearson correlation map, and they look very, very similar. Exciting. This is kind of what you would expect, just by knowing what we know from the way that the 3D genome architecture and species evolve. And they're similar, in the maps on the eigenvector side. And we're at that point, okay, this looks like the fidelity of the compartments is kind of preservation is high enough that maybe we are able to pick up cell-specific patterns, and indeed we do. So we generated a whole panel of tissues for the Asian elephant, and again and again comparing the contact maps and the pattern, the compartment patterns, we see that the Asian elephant skin is closer to the mammoth skin than to any other Asian elephant tissues. So that's super exciting, which basically means that we really have first of its kind, high resolution data of compartments. And of course, again, through all of the efforts that have been done, we know how to read that data off for things like gene activity. So we generated this genome-wide data to basically tell which compartments are open and closed, which genes are there, basically giving us the first of its kind of map of gene activity in the woolly mammoth skin. Naturally, we compared it to the that in the Asian elephant. More, broadly speaking, they're very, very similar, which is again something that you would expect given what we know how species evolve. Of course, we were interested in annotating the differences, and indeed, this is something we do in this work. I don't have time, and again in the 20-minute talk to go into details, just listing this there as, something that maybe you will want to check out, and just going to say that interestingly, a lot of the genes that got highlighted had something to do with the hair and just skin maintenance, and this is sort of indeed kind of the thing that you would hope for, because it is a skin sample, and the mammoth is indeed woolly, which is differs it quite dramatically from the elephant. So we were excited to see those differences. All right, well, and so yeah, clearly we can see compartments, and I'm sure that you won't be shocked by, seeing where I'm trying to go with this, right? Like, well, how about to try to go even further, and what about loops? So, right, well, at this point, we'd love to look at the loops. The only problem is that usually when you want to look at the loops, you really need like a few billions of reads, because, to see those point-to-point, really, really fine interactions, and we didn't have those here. So what we did instead is we annotated loops in an elephant, and we tried to employ something that's it's a really, really smart trick that has been pioneered in 2014, is this aggregate peak analysis. So, right, what you do is, if you know where you expect the peaks to be, what you can do is like locally pile up the pieces of the map on top of each other and just analyze them in aggregate. And this is what will happen if you take a typical extant species, right? Here's the elephant, you annotated the loops here. If you pile them up on top of each other, you see this like very characteristic enrichment over here. So great, this is sort of the tell sign that the loops are there. So what about the mammoth? Right, if we try to do this individually in the corresponding positions in the mammoth, the map is too sparse, you won't have a chance to see individual loops, but in aggregate, look at this, here they are. So that's, we're super excited to see this picture. And indeed, so excited we decided, maybe we can play this game even more ambitiously, and not just look at the loops that we picked up from the elephant data, but actually try to trace the loops, in the mammoth through something like 105 million years of evolution, and just take basically a list of loops that we have as humans in our skin, lift them over, find the corresponding positions for the anchors in the mammoth, and pile those positions up. And again, you kind of see this enrichment, which was something that was very exciting for us to see. So, indeed, this kind of very confirmed to us that it seems that we at least can continue to zoom in all the way down to loops, basically gives us kind of like a scale all the way from like the whole piece of the mammoth skin on the scale of meters all the way down to loops, something on the scale of 50 nanometers. And we thought that really we seem to be looking at something special, it's something different, not the kinds of fossils that people have been talking before, but, and sort of to try to highlight the difference, we tried like experimenting with this term fossil chromosomes. Again, like people, these are non-mineralized subfossils, but it's just too much of a mouthful. So, I'm just going to leave you with fossil chromosomes for now, and give it up to Eris to